The next speaker is Josh Denny.
Well, good afternoon. It is a pleasure to be here and hear about the incredible things that are going in the UK Biobank as a follower for many years, and talk about phenotyping from a wide-angle lens, as John stated. So, starting this problem, thinking about maybe first as genomics and how we thought about associating variables with those two as predictors, we started phenotypically in history with things like survey instruments and exams and careful studies. And one of my foci has been, how do we focus and how do we use electronic health records as a wealth of information about the clinical trajectory of disease? And then, there are many other types of phenotypic information that we are collecting in our cohorts that become powerful tools to help us disentangle and personalize disease treatment over time. And so, focusing most on electronic health record, I want to give you one example of two patients that were at, John mentioned I used to be at Vanderbilt University, two patients at Vanderbilt University and their blood pressures, and the simple question of who has hypertension. And anyone who's treated patients know that there's a lot of variability, and it's of course defined clinically by blood pressure measurements. But if you use common thresholds, there are so many things that influence this that you can get, false results if you just use that. And patient one, who has elevated systolic and diastolic blood pressures, doesn't have hypertension, but came in for a bone fracture, which caused pain and raised their blood pressure. And patient two actually has hypertension, but is incredibly well treated, as you can tell here. And I wish all the patients I had hypertension had, blood pressure readings that looked like this. And so, in fact, using just the blood pressure with even complicated algorithms alone is not your best predictor. It's much better to include things like medication, diagnoses. Diagnoses by themselves work better than just blood pressure alone to pick who has hypertension. So because of that, people apply algorithms. And this is an early study, one of our first discoveries in the eMERGE network, that found something new using just electronic health records to phenotyping. And our algorithm included billing codes, lab tests, temporal identifiers between those lab tests, the billing codes, medications that were contraindications, procedures that were contraindications, and produced an algorithm that was very accurate and found, identified a signal in chromosome 9, which is associated with hypothyroidism. This has been replicated in many cases. This was, at the time, a new discovery. And so what you find is the evidence of disease is in many parts of the record. And this probably doesn't surprise anyone. It's not just about disease subtypes, though, and longitudinality of disease or what happens in progression of a disease. It's just even the record of the disease itself can occur in different parts. And you don't need to read those different parts of that Venn diagram, but the different colors represent different parts, from billing codes to medications that are highly specific for the disease to note indications. And you can see that the patterns look different for different diseases. And so when people create an algorithm to identify these and create highly research-grade phenotypes, they typically use multiple components and combine them in different types of logics. And you can see that billing codes are the most commonly used things, and a number of algorithms that have been posted on a website called PKB, and medications under there. And then some people are using natural language processing on cups of narrative notes and other types of information there. And so there are many ways to go from the process of creating something that's complicated using Boolean logic and the rule-based algorithm, hypothyroidism, to something that is faster and more high throughput, as Naomi indicated. How do we simplify this process? And so one of those would be approaches like FIWAS, which we heard earlier. Typically those rely on some sort of aggregation of billing codes, and on the right shows some of the initial groupings of ICD-9 and ICD-10 codes into a common phenotype nomenclature and backbone, and how they can also group to control conditions that you'd want to exclude from a control group potentially as potentially overlapping disease conditions. And so usually there are ways of aggregation. Other people have applied other approaches using domain and controlled terminology hierarchies. Machine learning approaches can be used as well to group codes related. There are, of course, other types of phenotypes. We talk about this a lot. Surveys, using natural language processing. You can do the same sort of FIWAS approach across laboratories on procedures. And, of course, a lot of interest has been focused around artificial intelligence, machine learning approaches, and how we can even move beyond just clustering approaches, but data-driven clusters and such as deep neural nets can give us on what is happening in different conditions, and maybe what those topic modeling or other approaches could identify as clusters within a disease or across a wealth of diseases, and finding hopefully predictive analytics for a patient population. And so one example of a FEWAS, what I like here is this is grouping across atrial fibrillation and a genetic risk score. And what this shows you is when you use a genetic risk score for atrial fibrillation, there are a number of conditions that show up that are, clinically we know related to the same patient population that is at risk for atrial fibrillation. But what you can do is you can condition also this analysis and rapidly test, are these other associations potentially genetic pleiotropy, or do they represent, associations that occur with the underlying conditions? And it's very rapid to do this kind of analysis with something like a FEWAS, and you see on the right, if you adjust for patients that have cardiac arrhythmias, those other associations go away. Another thing that using something like this as a backbone can help you do is combine cohorts. So in the US, we used ICD-9, a modified version of ICD-9 until 2015. Then we moved to a modified version of ICD-10. The UK uses ICD-10. And these have different numbers of codes in them and slightly different architecture to them. So in this case, this was an analysis that looked at 23 hereditary cancer genes, looked at those that had pathogenic or likely pathogenic variants in those genes that you can see on the x-axis, and then looked for associations by FEWAS with these different conditions. It was started initially in the eMERGE cohort, and then combined a Vanderbilt clinical cohort, and then we were able to add UK Biobank to it, and really in the matter of a couple of weeks of pulling those codes in, mapping into a common architecture, and then doing those associations. And in this analysis, we replicated 38 of the known ClinGen gene cancer associations and a number of the overall associations mentioned in OMIM, and identified some new associations. And it's really made possible by having those sorts of interlingua between some of these coding vocabularies to speed up that kind of association analysis. So as John mentioned, I now lead the All of Us research program, which is, I will say, a sister program to the UK Biobank. We've learned a tremendous amount from the UK Biobank. We borrowed a number of survey questions from the UK Biobank to try to maintain harmony and to facilitate cross-analysis over time. We launched in 2018, and we've enrolled more than 557,000 participants. People participate in different ways across the U.S. So amongst those, about 400,000 have completed the initial steps of the program. And by virtue of our electronic health record data that participants donate, there's quite a bit of longitudinality to that data, which of course is seen in a great way through the UK Biobank as well. And so we have about 40 years across our EHR data. People are sharing Fitbit data and other sorts of data as well. And so about 12,000 participants with Fitbit data that goes back up to about 10 years also. And you can, of course, see the survey data is limited to when people started enrolling. A big notable point of the program is we have intentionally focused on enrolling a diverse population and reflecting, but enrolling amongst underrepresented populations in the U.S. So about 50% of our population represents diverse genetic ancestries or by race and ethnicity as they enroll, and over 80% are underrepresented by medical research. And so as these data come in, they come in the U.S. system across many different individual data providers. So our medical record systems come across 16 different vendors, probably actually more than that. They get harmonized into a common data model, and using that common data model, which we use OMOP as our data model, that enables much more rapid assessment across those phenotypes. So if you put in, for instance, cancer, synonymy to malignant, plastic disease as the preferred term, and then it gives you all the SNOMED, ICD-9, ICD-10 codes for those conditions marked together, which makes it more, facilitates the analysis. And so to give this into practice, this was my last PhD student at Vanderbilt. He defended after I joined the NIH, but his analysis started with a process of using gene expression data and identifying drugs that might lower cholesterol, and then finding those candidates and testing them in the Vanderbilt EHR. And these are some of the results. Probably not surprisingly, most of the drugs that lower cholesterol that he found in his hypothesis-free approach are known cholesterol-lowering medications. But the question that came in this drug response phenotype, which is looking at exposure, a number of things to refine the phenotype, the drug, and then looking at the lab and lab change afterwards, was this took 3 years. Can you replicate these data, and does this translate anywhere else? And so, he was able to take this take his code from Vanderbilt, which has the same data model, and a number, hundreds of millions of participants across the world are in this data model, and replicate it in all of us in about a week from when he first applied to the resource to when he was able to generate a figure that looked like this, and most of the results align closely. And so we talked about different analyses and different components, and here's one on physical activity using PheWAS approaches as well, and you can see the association between physical activity and a number of different phenotypes, and then the association between number of steps and development of conditions like diabetes, hypertension, gastroesophageal reflux disease over time. And then finally, important thing that we're trying to make sure we enable is cross-analysis across the UK Biobank and resources like All of Us. And worldwide, we want to help facilitate this as well. And so this analysis, which is on our preprint server now, looked at plasma lipids in the UK Biobank and All of Us, and cleaned that phenotype in similar ways, and then tested what the results look like in a pooled analysis versus a meta-analysis. And you can see that you have similar effects in LDL levels, for instance, on statin and not on statin between the UK Biobank and All of Us. And then doing the analysis, both approaches worked well, and we replicate a number of signals across that are known in the association analyses. And so some closing thoughts. Our goals as those developing a cohort should be to make simple things easy and make hard things possible. I'm a big advocate for common data models and common technologies to increase the speed of what we do and interoperability, reproducibility, and science. And I think it's important that we also, as much as possible, collect raw data with those signals that we process as well, so that it enables emergent analyses such as with artificial intelligence and machine learning approaches. Thank you.